Hello students, today we will start with meiosis. In the last session we have studied cell division of mitosis, the phases of mitosis stages, prophase, metaphase, anaphase and telophase that we have seen in our last session. Now we will start with meiosis. What is meiosis? It is the process of the reproductive cells which divides into the 4 haploid daughter cells. Okay students, so we will see how this process will go and how it will be easy for you to understand. Okay, one thing you have to remember is that the meiosis, why the meiosis is done? Because the genetic diversity that we see all around is just because of the meiosis. Okay, and also one important thing to remember is each new cell of the meiosis which undergoes the meiosis carries genetic material and genetic information. Okay, and after meiosis what happens to it? The union of set or sample of this genetic material, what forms? It forms the new organism. Okay students, so the new how the new organism can be formed by meiosis, this can be understood by this now in this stages. Okay, so there are 2 cycles of meiosis, and first cycle is just like the mitosis. Okay, so here is the first cycle and this is the second cycle of meiosis. So in first cycle of prophase 1 we will say for that, here it is a cell, it has centrioles and this is a spindle fiber which has, the cell has nucleus, nuclear membrane, and this nuclear membrane starts to dissolve just like the mitosis here, and DNA, what the genetic material, that is DNA, that thickens in this process and chromosomes condenses, starts to condense. Okay, and after condensation what happens? The nuclear membrane dissolves and the chromosomes which were inside, they released from there. Okay, they get released, and in metaphase what happens? The centrioles which were in this prophase, they reached towards the ends or poles. Okay, and the spindle fiber get attached to the chromosomes here. Okay, and this chromosomes they are attached to the centromere. Okay, this is the centromere where they are attached. You can see in this diagram. Okay, they are homologous chromosomes. Homologous chromosomes means what? They are genetically, they are alike or they are like chromosomes. Okay, they are composed of the same genetic copies, means genes copies. Okay, so they are known as homologous chromosomes. They are lined up. You can see this line. These are lined up in a very similar manner. Okay, and in anaphase what happens? The centromeres are at the poles and this centrioles are at the poles, and these centromeres they are pulled by the spindle fibers, and the sister chromatids are there here, they get they stay together here. Okay, they stay together here. The sister chromatids, you can see here, they are together, and the centromeres are attached to the spindle fiber. They get pulled. Okay, and in telophase what happens here? The chromosomes which are at the ends, now they are just the nuclear membrane forms around them and the two different type of cells are produced. These two cells are produced here which are haploid. Okay, because each chromosome has two non-identical sister chromatids. Here see, this is non-identical sister chromatids. These two are non-identical sister chromatids. Okay, this was the end of phase 1 of meiosis. Okay, here we got two haploid cells which has two non-identical sister chromatids. Okay, now this cell here also one thing is there, the cell membrane also constricts, and by constriction the two cells are separated, and by this separation in prophase 2 what happens here? Here same process for prophase 1, prophase 2 also has the same process. Here homologous chromatids, these are the homologous chromatids, and these homologous chromatids separates but they do not duplicate. Okay, and the chromosomes condenses just like the prophase 1. Okay, the nuclear membrane starts to dissolve, and in metaphase here same process, centrioles are gone at the poles, spindle fibers are attached to the centromere here and they are lined up. Okay, chromosomes are lined up. This process is metaphase 2. Okay, in anaphase now cell becomes constricted at the center and the chromosomes which are attached to the spindle fibers, they pull the centromere and they got separated, and the sister chromatids which were formed, they are separated. Okay, and in telophase like that only the cell membrane again constricted to form other two non-identical cells. Okay, so these are two non-identical and these are also two. Like this here 4 non-identical cells are formed. The chromosomes here in this are non-identical. Okay, so they are called haploid, 4 haploid daughter cells. Okay students, they are newly formed gametes which are haploid. So here in this meiosis phases are completed, and what we get in the result, that is 4 haploid daughter cells. Okay students, so they are non-identical and their genetic material is not identical. Okay, so we all look unique. Okay, so this students, in mitosis mitosis helps us to grow, and meiosis is what we look unique. So students, you understood what is meiosis. Thank you students for watching.